Hi, it's Mr. Andersen, and welcome to the final AP Biology review video. I asked you to submit questions, and boy, did you submit a lot of questions. So I'm going to go through all of those. I would imagine this video is going to be very long, but I'll put some links so you can jump ahead to the parts that you're maybe interested in. And so again, I asked for questions, and so I got them. So I got questions from 39 different states, 20 different countries, hundreds of different students submitted their questions, and so I'm going to go through the most popular ones in this video. This is a neat comment I got from one group from Belmont, Massachusetts. Our AP Bio has watched so many of your videos. We'd like to thank you so much. You've helped us. That on our t-shirts we are writing, and I hope that's helpful on the back. Thanks for all the help. And so that's a cool comment, and I got tons of thank yous and comments which make all the work worthwhile, and so thanks for that. And so this is the table of contents. What I'm going to cover in this video is, number one, I'm going to show you a web page where you could go to and you could kind of find some of the answers to your questions if I don't cover them in the video. I'm then going to go through the top 10 requested questions, and so this starts with test strategies and goes through statistics. And then finally, I'm going to give you a bunch of you a good luck shout out. And so I haven't done this in YouTube, but I'm going to give it a try. I should be able to insert a link here for the top 10 requests and for the good luck shout outs. And so if you want to skip ahead to that, then you should be able to click on that. If not, sorry, you're going to have to do a little bit of scrolling. And so let's talk about the website. If you haven't been to my website before, that's bozemanscience.com. And I'm going to jump over there right now. So this is bozemanscience.com. It's going to have different pages for the different material. So if you click right here on AP Biology, it's going to show you links to all of my biology videos organized in order. There's got some supplemental material here. We've got some labs up here, so you can find more information on topics that you're interested in. You could also go on the left side down here to the search, and so if you have a question about operons, for example, you can type that in and you can find all my videos that are related to operon, including the operon. One other page that I just added is this. So if you go to bozemanscience.com/review, I've added a new page. It's kind of a hidden page. And so I've got all of the questions that I don't get to in the video, then you could answer those here. So I point you to different videos. I try to answer some of the questions as best I could. So if you don't find the video or the question you're looking for in the video, you may want to go here again, bozemanscience.com/review. Another place to look is to go to my YouTube site. So that's youtube.com/bozemanbiology. And if you go there, you can always search in the channel up here. So if you click on something like operon up here, in that search box, then it's going to find all the things related to operons, and that's going to be maybe a little bit easier way to find it. So whatever works for you, use the website, the hidden website, to find information that I don't cover in the video. Okay, the next thing I want to do is I want to go through the top 10 requested questions. And so these are the burning questions, the ones that a lot of people wanted more information on. So it was sometimes 30 people would submit questions in each of these categories. The first one was people wanted information on the statistics of the test, like what does it consist of, what are common questions, things like that. Next, test strategies, how people could improve their scores. They wanted to know that. Followed by more information on the labs, and I'll get to that. Next, cell communication, chi-squared test, and Hardy-Weinberg are always popular, and I'll work through a couple of sample problems. Next, people wanted to know more about water potential, photosynthesis, respiration, and finally statistics. And so I'm going to work through all of these, work some sample problems, and then we're done. I'm going to get to the good luck shout-outs. And so when you take the test on Monday, it's going to be broken down into two parts. Part one is going to be multiple choice and grid-in questions. You're going to have 69 total questions, and so those are going to be 63 multiple choice, and then the 6 grid-in, remember, you're going to put in numbers here and fill in the corresponding numbers below it. So these are going to be ones where you use your calculator, you use the formula sheet to answer them. That's going to be half of your grade on the exam. You'll have a break right here in the middle, and then you're going to have another 90 minutes. That 90 minutes is made up of two parts. You're going to have a 10-minute period of time where you read the questions. These are the essay questions. They're going to be 2 long form, and then they're going to be 6 short form essays. And so you'll get to read the questions for about 10 minutes. This is super important time where you plan out how you want to answer the questions, and then you're going to have 80 minutes to actually write, and you'll write that in a separate booklet. What do you need to bring for the exam? Remember pencils to do this part, and bring a blue or a black pen for the essays. As far as test statistics, I'm going to go through questions that I got from people and try to answer those. Number one, this is from Alex from South Dakota. Do you think the new and revised AP Bio tests will be harder than the past tests? I would say this: the AP biology tests are always difficult. They're designed to be hard and to sort out who really knows biology and who doesn't. And so since they're all curved, in other words, they'll put it along a bell-shaped curve, they're going to figure out standard deviation and figure out where you're at, it's going to be the same difficulty for everyone. And so it doesn't really matter that it's going to be maybe harder this year or harder another year. We're still going to have about the same number of 5s, 4s, 3s, 2s, and 1s. It is changing. In other words, there's not going to be a lot of questions that are just factual recall. There are going to be more questions where you're actually going to have to work to find the answer or do some application or interpretation. So you want to be prepared for that. Be ready to work, especially in the multiple choice. Next question is this. This is from Cooper from California. How much is each individual section weighted? For example, is animals and plants worth more than genetics and ecology? They've kind of changed the curriculum, and it's basically centered around these 4 big ideas: evolution, free energy, and this would be homeostasis, information, so this is going to be like DNA, genetics are in here, and then systems and interactions. And so the questions are going to be equally divided between those 4 big ideas. You could go to the College Board's website, and I'll try to put a link in the video description down below, and you could look at the whole curriculum if you really have time and see where most of these questions are coming from. If you've watched a lot of my video essentials, I center those videos around what are the important essential understandings that you should have. And so if you've gone through the curriculum, you should be ready to go. Next one, will there be only one correct answer for the grid-in, and will these be computer graded or by hand? That's a good question. So there will be an answer, but there's going to be a range at which your answer could be in. And so, for example, maybe the answer is a Q value in Hardy-Weinberg. The right answer, if you did it with all the significant digits, is like 0.3456. The value that they would accept would be maybe between 0.34 and 0.35. And so there's going to be a range, and you can make sure that your answer is somehow in there. And so it is computer graded, remember, the grid-ins. And so make sure you're bubbling in those appropriately. And I did find kids got into trouble if they rounded really quickly. You want to kind of leave some significant digits in so you don't get screwed up on that. Thanks for that question, Miss Churchill's class from California. Next, from Monica in New York, what percentage of each part would you need to get correct to get a 4 and a 5? That's really going to vary. And so depending on how hard the multiple choice questions are and the essays, I can't tell you. I would say this, that it's going to be harder than a conventional test. And so if you have a test in high school biology where you're always getting, I consistently get an A or an 80% or more, or an 80% or more, or 90% more, or 70%, it's designed to be harder than that. So there's going to be a number of questions that are very difficult. And so lots of times, historically, if you can get, over 70% of the questions right, you're looking at getting a 4 or a 5. Now that we only have 4 responses in the multiple choice, you might have to score a little bit better than that. And so there's no hard, fast percent you have to get right. We won't know that until you've all taken the test. Next one, Pasquale from Connecticut. How long should the first 2 response questions be? They're going to have multi-parts in them. So the long ones will have part A, B, C, D, sometimes E. And so they're designed to take you quite a bit of time. And so as long as you're working through each of those subpoints, on average, it should take you about 20 minutes to get through the long responses, and the short responses are going to be more like 6 minutes to get through all of them. And so you want to kind of keep yourself on pace. What I found with my own students is that generally they're pressed for time on the multiple choice. They'll be pressed for time on the grid-in, but a lot of the time they tell me at the end, everybody's kind of done writing, and the last few minutes are not people still working on the essay. And so I don't know, it'll be up to you depending on how fast you write, but I would put a lot of time and make sure you don't skip any question. Danielle from New York says, what happens if you get below a 3 on the exam? Do you give up your dreams of going into the science field? No way. If you're interested in science, you should study science. And if it doesn't go well for you, you might not have done a great job this year, but it doesn't mean that you can't go into a science-related field for sure. It means that you're going to have to work. You didn't pick up the material if you fail this that you should get from a regular biology class. But if you start over again, are really interested in it, I think you'll do fine. Next, Ariana from the U.S. said, could you go over any major diagrams and graphs we need to be able to read? It's a good question. I don't think I could do that because I obviously don't know what's going to be on the test. I would tell you this, that there's going to be tons of questions where you're going to have to do graph analysis. And so just get ready for that, making sure that you're reading the labels and reading what the question's asking for. It's going to be a lot of questions where you're going to have to analyze data And so be prepared to do that. And then you're going to have to use models. And so models, remember, are just graphical representations of a concept. And so even though you might get a question where you don't understand what this is right off the bat, just kind of work your way through the model, and then if you can apply some of the knowledge that you have, you might be able to get it. Next one, this says, what math concepts could be on the exam? Now, hopefully you've downloaded the formula sheet for AP Biology. If you just Google AP Biology formula worksheet, you'll find it. And it's going to have tons of information on it. So everything from the mean to standard deviation, chi-squared, pretty much anything on this sheet is fair game. There are going to be ones that maybe are more popular. I anticipate we're going to see some chi-squared and Hardy-Weinberg for sure. Maybe not one of the obscure ones, but you never know. And so you want to be able to answer any of the questions on that formula sheet. You've got a calculator, and so you got to be able to use that. Thanks for that question, Crystal from California. Next, Ellen from Washington said, what is the essential difference between this year's new exam and the previous exams? And so now we're moving into kind of strategy type questions. The big thing is that AP Biology used to be a mile wide and an inch deep, and what that means is we covered a huge amount of material. We spent, a month just going through all the different phylums and classification, this diversity of life. And so they've removed a lot of that, but they want you to go into more depth. And so as a result, you're going to have to do more application of the knowledge that you have. And so there aren't going to be just questions where, boom, I can look at it and I know that's the right answer. That's the thylakoid membrane. What they want you to do is take the knowledge and then the practices of science. So how we can use a model or how we can use the scientific method or experimental design to answer questions. And so really you should feel a little less stress where there's not a huge amount of material you have to know. You just have to be really thorough in what you do understand. Next one, Mary from Florida says, what should we do the weekend before? You can still study. I mean, this is a test where if you don't understand photosynthesis, you better figure it out this weekend. The night before, you really should get a good night's sleep, but Saturday, Sunday, you can do lots of study and figure out what you don't know. If you haven't taken a practice exam, that's what I would do first. Figure out what you don't know and then go study that. How do I answer all the multiple choice questions when they're so long? There's so many details I get lost. Remember, some of the questions are designed to be super hard, and some are designed to be super easy. And so if you get an easy question, don't overthink it. If you get a hard question and you're spending, 2 minutes on a multiple choice question, I would encourage you to skip it, and then the last minute, go back and fill in everything. And if you have to guess, do so. There's no penalty for guessing. And so make sure you go after it. Next one, how should I prepare for the essay question, says Rachel from Hong Kong. For me this week in my class, what I was working through is these what are called direction words. And so on the essays, you're going to have these questions show up quite a bit. And so it's not about the content of biology. It tells you how they want you to answer the question. And so if you don't know what these 8 things mean, explain means to make it understandable. Discuss means to talk about various aspects of a concept. Define is to give meaning. Describe is to characterize or use words to explain it. Justify means to prove. Compare means similarities. Contrast means differences. And if you don't know all the good parts of a graph, you may want to figure out what these words mean, because some of these are what I call danger words. If you see explain, you're not just going to answer that with one word or one sentence. They're looking for a number of different things. You really want to make it understandable. If they just say discuss, that's an open-ended question. And so it's important that you understand what these direction words are. It's going to help you at least forming the way you want to write your essay. Emma from Rhode Island says, if you're stuck on a free response question, is it better to leave it blank or try to write something even though it might be way off? Don't worry about wrong information. Don't worry about spelling. Don't restate the question. You're not going to get points for that. In AP Biology, they want you to write down what you know, and they're going to grade you on what you know. And so lots of times you'll have essays that are really difficult. And so across the country, out of 10, maybe the average is 3 or maybe 2. And so if you can get any points, that helps you. And so I found the only time I see kids really bomb out is when they did well on the multiple choice, but they say, I didn't understand what one essay was and I just skipped it. Don't ever do that. Battle for every point on the essays. Next, Kaki from California says, do a single grid-in question and a single multiple choice count for the same? Absolutely. And so if you're stuck on a really hard grid-in question, don't spend 5-6 minutes doing that and not answer some of the multiple choice questions. And so a grid-in is worth just as much as a multiple choice question. It doesn't mean skip them all. You should try to do all of them. But it does mean don't spend tons of time there and lose out on the other side. Okay, next thing I want to talk about are the labs. There are 13 labs that you should be familiar with in AP Biology. I put together 2 videos, and I'll try to put links here for part 1 and part 2. These are on YouTube, and you can go through and look at the 13 major labs. They're not super detailed, but at least they'll give you enough information so if you're taking the test, you can answer those questions. Okay, number 4 was on cell communication. I'll put together I have put together a video on cell communication. There's another video I've done on quorum sensing in bacteria, and so you may want to consult those. People get nervous about cell communication. It's fairly simple. If we have one cell and then we have another cell and we want to communicate between these two cells, generally what we do is we send a chemical. We send a chemical message, and that message is going to move to another cell. It might bind to a protein on the surface, or it might actually move into the cell itself. And so what are some examples of this? Well, in bacteria, they'll communicate with each other using what's called a autoinducer. An autoinducer is just a message between bacteria. In us, if it's very close, we'll use what's called a neurotransmitter between nerves. If we're talking about endocrine system, we're going to be hormones that we're using throughout our body. And sometimes, like in the immune system, we're just going to use a chemical. So we're going to just use a shape of an antigen to figure out how we communicate between cells. And so again, I would encourage you to watch the video on cell communication if you're stuck on that. But it's really just a way that cells communicate with one another. Okay, next thing I want to do is I want to go over some chi-squared problems. And so this is one that gives a lot of students fits. And so this is the one that I gave to my students on their AP exam. So this is their final. So in pea plants, the purple allele is dominant to the white allele. Gregor Mendel performed a cross between 2 purple-flowered pea plants. When planted, the 236 seeds that were produced from the cross matured into 164 plants, 72 plants with white flowers. Calculate the chi-squared, so we know it's a chi-squared value kind of a question, for the null hypothesis. And so for me, if I'm ever trying to answer a genetics question, the first thing is I want to talk about what I've been given. And so they say here that the purple allele is dominant. And so what I would write is big P equals purple. And then what else did they tell me? It's dominant to the white allele, and so little p is going to equal white. So I know that. What else have they told me? Well, they're telling me that the parents were both purple and that they were heterozygous. Okay, so that means I'm going to have to do some kind of a Punnett square. I'll get to that in just a second. But this is a chi-squared, and so a chi-squared, you know it's generally a chi-squared if they give you values. And so they're telling me that the number of purple plants were 164, the number that were white were 72, and this is the total, 236. So in general, for me, when I'm doing a chi-squared test, I like to organize it like this. I write down the observed values, and I'm going to write those across. So our observed values, we had 164 of the purple, and then we had 72 of the white. And so what I have to do is figure out the expected values. Expected values, we're going to have to use a Punnett square to predict that. And so they're saying that the parents were both heterozygous. And so I'm going to just write out a Punnett square for those parents. And so that is big P, little p, and that's going to be crossed with big P, little p. And so I'm going to write this one as be big P, big P. This one's going to be big P, little p. This one's going to be big P, little p, and this is going to be little p, little p. Okay, so what would Mendel predict? Mendel would predict that these 3, 1, 2, 3, are all going to be purple. And then how many are going to be white? Just 1 of 4. And so another way to say that is it'd be around 75%, or 3/4 are going to be purple, and 25% are going to be white. Okay, so now I got to figure out my expected values. There's a total of 236 seeds, and so what I can do is I can take 236 times 0.75. And if I do that on my calculator, let me grab my calculator for a second. So I'm going to take 236 times 0.75, and I get 177. And so how many, if Mendel's right, my expected value here should be 177. Now let me figure out my expected values over here. So we could say this is white and this is going to be purple over here. And so how do I figure that out? Well, my total again is 236. This time I could take it times 0.25 because that's going to be how many are white. And so if I take 236 times 0.25, I get 59. And so what is the chi-squared? Chi-square test is going to be way up here. And so chi-squared test, this is just the symbol for chi-squared, but it's going to be the summation of O minus E squared over E. And so now that I have all of my O's and E lined up, it's really easy to solve this problem. And so I'm going to write my observed values. So my observed values is going to be 164 minus 177. And I'm going to square that, and that whole thing is going to be over my expected values, which is 177. I have to do that for my other side as well, and so I have to do that for the whites now, and so that's going to be my observed value, which is going to be 72 minus 59. I'm going to square that, and then I'm going to put that over my expected value, which is now 59. And so if I do this kind of in my head, 164 minus 177 is negative 13. And if I square that, that's 169 over 177. And so if I do that in my calculator, 169 divided by 177, and I get 0.954. So for this first one, I get 0.954. So that's going to be for the purple flowers. That's going to be my chi-squared value. Now I'm going to do my white ones over here, so that's plus. So this is going to be 72 minus 19, so that's going to be 13 squared again, so that's 169. This time it's over 59. And so I'm going to take 169 divided by 59, and I get 2.86. So 2.86, and that's plus 0.954, and so I get 3.81. And so what is my chi-square value? It's going to be 3.81. Okay, so what does that mean? Well, let's look at how many degrees of freedom that we have. And so how many degrees of freedom do we have? Well, there are two different phenotypes. We've got the purple phenotype, and then we've got the white phenotype. And so how many degrees of freedom am I going to have? I'm just going to have 1 degree of freedom. So that's going to be in this column. And how do I get 1 degree of freedom? It's n minus 1, where n is the number of different types I have. So I have purple and white, and it's going to be minus 1. So it's going to be 1 is going to be my degrees of freedom. And so what's my critical value? 3.84. And so we're really close to that, but we haven't exceeded our critical value, and so we'd still have to accept our null hypothesis. And so we could say that Mendel's right, that this is predictive, at least in this quick Hardy-Weinberg. Okay, let's go to chi-squared. So this is, again, the same one I asked on my final. And so what do we got here? Or it's like a one I asked on my final. So in a population of snakes, the allele for brown skin is dominant to the allele for red skin. An invasive hawk targets the red-skinned snakes. When the hawk is removed, only 8% of the remaining snakes are red. And so in Hardy-Weinberg, what I like to write out is the whole thing. So this is p squared plus 2pq plus q squared equals 1. And then what's the other part of that? It's going to be p plus q equals 1. And so what do I know? Well, I know that the brown, so I'm going to say brown, is going to be dominant, and then little b is going to be red. So that's going to be recessive. And so what did they tell us? Well, they told us when the hawk is removed, 8% of the individuals are red. And so this is where I pause. I mean, have they told you Q? Have they told you P, P squared, Q squared? What have they told you? Well, in almost every one of these problems, they're pretty predictive. It's going to tell you Q squared, because these snakes, we know what their genotype is. We know that they have both of those red recessive alleles, and so they're telling me Q squared. So Q squared equals, I want to make sure that I write this not as a percent, 8%, but I write it as a decimal value, so that's going to be 0.08. And now I can solve for my Q value. And so my Q value is going to be 0.08, and I'm going to take the square root of that. And what do I get? I get 0.282. So that's going to be 0.282. And once you get Q, you should immediately figure out P. And so what's my P value going to be? It's just going to be 1 minus 0.282, and that's going to be 0.718. Okay, so now I have P and Q. Once you have P and Q, you can answer almost any question. So what are they asking for? So if this is in Hardy-Weinberg, what will the frequency of the brown skin allele be for the next generation? Well, we've already figured that out. That's going to be right here. So that's going to be 0.718. And since they're asking for an allele frequency, we should just leave it as a decimal value. And then they say if 1,000 snakes are in the next generation, how many brown-skinned snakes will be heterozygous? Well, now we're going to use this right here, which is going to be 2pq. So we're going to take 2 times p, which is going to be 0.718. And then we're going to take that times q, and q is going to be 0.282. And so if I do that, let me use my calculator. So I get 0.404, or 0.405 actually, 0.405. Okay, so that's going to tell me the decimal or the percent of individuals that are going to be heterozygous. So how many are they asking for? 1,000 snakes. And so if I take 1,000 times 0.405, what I'm going to get is around 405 snakes. So 405 of the brown snakes are going to be heterozygous. And so again, for Hardy-Weinberg, I would always write out the equations, write what you're given, and then generally they're going to have to give you one of two things. They're going to have to give you the q-squared value because you know the genotype of those, or they might just straight up give you the allele frequencies, and then it's going to be a little bit easier to solve that question. Okay, next one. Next one's going to be on water potential, and this is a really common question I've got in class as well. So water potential is a little confusing, and I think the symbols are somewhat scary. Oops, let me go back to that. And so on this one, what we're looking at is psi, and psi is equal to two things. It's going to be equal to the pressure potential plus the solute potential. And don't worry, these are all written on the formula sheet. And so what does that mean? The overall water potential is going to be, it's actually based on many other things, but it's the pressure potential and the solute potential. And so what does that mean? Well, let's make a little beaker here. And so we've got a beaker that's filled, let's say, with distilled water. So we've got distilled water in it. And then let's put a cell on the inside. Okay, so this is distilled water out here. What's the water potential of distilled water? It's always 0. So that's going to be 0. But let's say we have a cell here, and this has some solutes in it. So it has some stuff in it. And so if we ever add stuff inside a cell, that's going to take the solute potential and it's going to drop it. And so it's going to be a negative value. And we'll get to why in just a second. And so what happens? Well, if you look out here, the water potential is going to be 0. If you look inside the cell, what's going to be the water potential? It's less than 0. So think of water potential, the way I think of it, as the potential energy of water. Like, where is there a lot of water? There's going to be way more water out here than there is in the cell. And so where is water going to flow? It's going to flow inside. And so it's flowing always towards a lower water potential. And so since there's still a difference in water potential, water's going to keep flowing in, and water's going to keep flowing in, and what's going to happen to the cell? It's going to get bigger and bigger. If this is a human cell, it might lyse. But let's say it's a plant cell. What does a plant cell have that a human cell doesn't? It's going to have a cell wall around the outside. And so as water starts to flow in, it's just like a balloon. As it starts to fill up, the balloon's going to start to exert a pressure, and it's going to try to push that water out of there. That's the pressure potential. So solute potential is always going to be 0 or a negative value. Pressure potential is always going to be a positive value. And so that's trying to push the water back out. And so eventually what happens is it's going to reach a balance in a plant cell. And so water's still trying to flow in, but the pressure is pushing some of that water back out. And so that's going to be overall water potential. Why is this important? Well, it's really important. So if we look at a tree is going to have roots, and those roots down here are going to have stuff in it. And so what is the water potential? Well, the water potential outside is going to be closer to 0. It's going to be close to distilled water, but it's going to be much lower here. And so where's the water going to flow? It's going to flow in here. It's going to go through the xylem. It's going to work its way up to the leaves, and eventually it's going to evaporate off the leaves. And so where's the lowest water potential? Lowest water potential is going to be right here. Where's the highest water potential? Right here. And since trees lack a true circulatory system, it's just water potential that's getting up to the top. Now, one other thing I should mention with that is how do we calculate solute potential? Solute potential is negative ICRT. And so what does that mean? Well, that's always a negative value. And so if I were to draw a cell down here, let's work through the things. So first of all, C is going to be concentration. And so the more stuff I put in the cell, the more concentrated it is, that's going to increase this concentration. What's that going to do to the solute potential? It's going to move it down since this is a negative value. So again, by adding more stuff, that's going to increase concentration and it's going to decrease the solute potential. This R is a constant value, and so it's not going to change. So I'm not going to worry about that right now. It's written right on your sheet. What is T? T is equal to temperature, but it's going to be in Kelvin. So you'll have to remember the number of degrees Celsius plus 273 to figure it out. But again, if we increase the temperature as the molecules are moving around in here more quickly, there's more area for water to move in. And so again, if we increase temperature, what happens to our solute potential? It goes down even more since it's a negative value. And so that's going to allow more water to move in here. What's the last thing? We haven't talked about I. I is going to be the ionization constant. And so what does that mean? Well, what does it mean to ionize? If I take sugar, so if I say, let's just take some glucose, C6H12O6, and I put that in water, it's going to stay together as this sugar molecule, as this hexagon. And so it's just going to dissolve in the water. But if I take something like sodium chloride, so if I take salt and I put that in water, it's going to quickly break apart into sodium ions and chloride ions. And so what we've really done here is double the stuff that we're putting inside the cell. And so if you ever have salt, the ionization constant is going to be 2. And if you ever have sugar, the ionization constant is going to be 1. And so what happens if we add these ions? Again, that's increasing the ionization constant, and that's going to decrease solute. And so again, the equations are kind of scary, but as long as you know that as solute potential goes down, water wants to flow in more, and then you just know how to solve the problems, it's really not that scary. Okay, next one then is going to be on the free energy, kind of. And so this is the photosynthesis and respiration. So free energy, remember, is equal to delta G. And so delta G is the change in free energy. And so if we were to put free energy on this side, so if we put free energy like this, these are the reactants, and then these are the products. And if I were to show you a little diagram like this, and this is time going across, so this would be like respiration. So our reactants have higher energy than our products, and so this is an exergonic reaction. If we were to, let me try a different color. So if we were to do a different color here and do photosynthesis, it's going to start with a lower potential, and then it's going to end up with higher potential. And so this would be an endergonic reaction, or a positive delta G. And then this one right here would be a negative delta G. But a common question I got was, could you talk about coupled reactions? Well, sure. It's this right here. You should be able to identify these equations. If you don't know it, please learn these. And so this is going to be glucose plus oxygen makes carbon dioxide, water, and energy. This is going to be cellular respiration right here. And so this is going to be an exergonic reaction. And so what we're doing is we're releasing the energy that's found in the glucose, and we're tying that to the formation of ATP. And so this is going to be up here an exergonic reaction, and we're coupling that with an endergonic reaction here. And so where is this energy going? That energy is going to make ATP. And so likewise, what's this down here? This is photosynthesis. So we're storing energy in the glucose, and then we can eventually use that glucose, if we're talking about a plant, to do respiration, or we can eat the plant and we can do respiration on that. And so it's really important that you understand that the free energy in our planet is all coming from the sun, and we can use that free energy, or we can convert it into another form. Okay, so another common question I got was photosynthesis. Could you go through the steps of that? And I sure would love to. Photosynthesis is made up of 2 parts. We've got the photo part and the synthesis part. Photo always stands for light, and then synthesis stands for the making. And so this over here is going to be the Calvin cycle. So it's named after Melvin Calvin. And so what I'm going to draw down here is a chloroplast. And so a chloroplast is going to have 2 parts to it. It's going to have a thylakoid membrane over on this side, and then over here it's going to have what's called the Calvin cycle. And this takes place out here in the thylakoid. These are going to be the stacks inside the chloroplast, and this out here is called the stroma. And so what's going on in the light reaction? Well, we need light, and so light is going to come in, and it's going to strike a photosystem that's going to be in here in the thylakoid membrane. What is it using that energy to do? It's using that energy to, first of all, be passed down an electron transport chain. So if I were to draw that, what does it look like? We have this photosystem right here, and so this is the light coming in, and so what that's going to do is get electrons excited, and it's eventually going to pass electrons down an electron transport chain. And so what does that mean? As they're moving down this gradient, it's going to end up pumping H plus ions to the inside. And so this over here is called photosystem 2, and that's because it was discovered second. What happens to the electrons? Well, the electrons, as they move through this, they're going to pump protons to the inside, and then it's eventually going to go to photosystem 1. What is photosystem 1 going to do? It's going to take that energy and it's eventually going to create what's called NADPH. And so NADPH is carrying electrons to the Calvin cycle. It's going to be one of the things that's going to be created in the light reaction. So this is NADPH. What is the other thing that's created in the light reaction? Well, let's keep watching that. So as this moves down, we're going to pump protons to the inside. So we get more H plus, more H plus. And what eventually happens, and I'm running out of space here on my diagram, is that eventually that's going to run through an enzyme called ATP synthase. And so as the protons run through here, that's going to generate ATP. And so ATP and NADPH are the two things that are made in the light reaction. Now, where are the electrons coming from? The electrons are actually being donated by the water. And so the water, or the H2O, is donating its electrons for the electron transport chain. What is it producing? Well, the protons are going to be the ones that move through the gradient. And then finally, we've got oxygen. And that O2 is the oxygen that you're breathing in right now. So in the light reaction, remember, what are the big things we need? We need a little bit of light. What else do we need? We need H2O. And what are we going to produce? We're going to produce some oxygen. That's the oxygen you're breathing right now. What's over here? Well, this is the Calvin cycle. And so in the Calvin cycle, what are we taking in? We're taking in carbon dioxide. Now, carbon dioxide in the Calvin cycle is eventually going to be used to make something called G3P. It's a 3-carbon molecule. It's got high energy. Where's the energy coming from? It's coming from the ATP and the NADPH. What happens when they've dropped off their energy? They're headed back to the light reaction. So this is photosynthesis. Again, this is going to take place in plants and other producers, and we can't do it. And so that's how they make G3P, and eventually they can use it to make sugar. So we don't do that, but we do cellular respiration. So what is cellular respiration? Cellular respiration, let me quickly draw a chloroplast, a mitochondria. And so this is going to be a mitochondria. So what are the 3 steps of cellular respiration? Well, the first step is going to be called glycolysis. What happens in glycolysis? We're going to take in glucose, which is a 6-carbon molecule, and we're going to break it down into 2 molecules of pyruvate. Okay, so these are 3-carbon molecules. As we do that, as we go from 1 6-carbon to 2 3-carbon molecules, this is starting with a 6, remember. What we're doing is we're releasing some of the energy that's locked in that glucose. And so we're going to make a little bit of ATP here, just to make 2 net ATPs at this point. So where's the energy going? Well, the energy at this point has been converted into something called NADH. And so what is NAD doing? It's carrying electrons, and it's eventually carrying electrons to the electron transport chain. So what happens to that pyruvate? That pyruvate is going to diffuse in through the membranes of the mitochondria into what's called the matrix. It then becomes acetyl-CoA, and then it enters into what's called the Krebs cycle. The Krebs cycle, don't confuse it with the Calvin cycle, but what's happening is that acetyl-CoA, as it goes into the Krebs cycle, is eventually going to convert these carbons down into carbon dioxide. So we're going to make CO2. So that CO2 in our atmosphere is coming right here from this Krebs cycle. What do we create in here? We create a little more ATP. So we're going to make 2 ATP. What else do? We're going to send that energy from the Krebs cycle. That's going to go to NAD, NADH, and it's going to also go to FAD. What are they carrying? They're carrying high-energy electrons to the electron transport chain. So this is the last step right here. What happens in the electron transport chain? Those electrons from NAD and FAD are going to be used to pump protons to the outside of that membrane. This looks a lot like photosynthesis, and as these things flow back in through ATP synthase, we're going to make a bunch of ATP. So how much ATP do we make here? Sometimes 32, sometimes 34 ATP. And so we're going to make a whole bunch of ATP at the end, and that's because we're harnessing all the power that was found in that glucose molecule. What I've just described is aerobic respiration. What's the aerobic part? Well, the oxygen. So the oxygen at this last point is going to be the electron acceptor. So as those electrons from NAD and FAD go through the electron transport chain, they eventually will be accepted by oxygen. And what's that going to create? It creates something called water. And so understanding where the reactants are and the products are. So where's the glucose? That's our food. What else do we need for cellular respiration? We need oxygen. And what does that oxygen do? It's the final electron acceptor. What are we making in cellular respiration? Again, we're making water. What else are we making? Carbon dioxide coming from the Krebs cycle, and then we're making energy in the form of ATP. Okay, last one then is on statistics, and so a question I kept getting was, what kind of questions could they ask us in relation to statistics? And the right answer is they could ask you anything. And so they could ask you to do standard error. They could ask you to do standard deviation. I'm not thinking that they'll give you a bunch of questions in standard deviation because this takes a while, but you should know conceptually what it is. They could ask you to do the mean. I hope you know what that means. That's just the average. And then they could ask you to do chi-squared. So they could ask you to do anything here. Some things that sometimes trip up my students, they forget what n is. So n is going to be the size of the sample. And so if I were to say, here's the data, I've got 10 and 9.2 and 8.8 and 3.3 and 4.9 and 10.1. So if these are my data that I've collected, how would I figure out the mean? Well, I would figure out the mean by simply adding all those up and dividing by the number that I have here. And that number that I have here is going to be equal to the n. So that's the size of the sample. And so if you want to figure out, standard deviation, what you're going to do is you're going to figure out the mean, and then you're going to find the average distance from the mean. We square that, and it's going to be over n minus 1. Take the whole square root of that. I think that's going to take a long time to do that, and so I don't anticipating them giving you a question like that, but you never know. Okay, that was quite a review, and so I hope it was helpful to you, and if you skipped over that, that's probably a good sign. It means you're ready for the AP exam. One thing I did on my form is I asked you if you want me to wish you good luck on the exam, just check a box, and I would love to wish you good luck. And so that ended up being a lot of people. And so we're going to go through this list. It's going to take me a little bit of time, but I really appreciate all of the thoughtfulness you put into your comments. Thanks for that. And I want to give you a good luck wish. I want you to do well on the exam. So really best of luck. And so let's start with Alabama. From Alabama, we had Joey and Lila. Good luck on the exam. I hope you do well. From Arizona, we had Tyler, Mrs. Thomas's AP Bio class, Nico, Sasha, Maryvale High School AP Bio class, Fran, Matthew, and Vita, Jesse, Aaron, and Michelle, and Shiv, and Kevin. Congratulations on finishing the class. Good luck, Arizona. In Arkansas, we had McKenna and Miss Walchuk's AP Bio class. Good luck. From California, we had Alan and Andrew and Angelica, Amelia, Elizabeth, Isa, Donald, Deanna. We also had Dante, Danny, Cooper, Clarissa, Cynthia, Marin Catholic AP Bio class, Christina, Christian, Bradley, Ashley, also Ariana and Victoria, Annam, Aliana, Hannah. Also good luck to Grace, Esther, Emma, Rigetti AP Bio class, Crystal, Krish, Kevin, who else? Kelly from California, Katie, Kaki, Juju, Joseph and Jonathan, Jimmy from California, Jennifer from California, Jaihi, Isa, Miss Schlegel's AP Bio class from California, Mr. Didden's AP Bio class, and Mr. Buck's AP Bio class. Also good luck to Mia and me, and also Matthew and Marianne and Mark and Liney and also Leona and Savannah and Sarah. Also good luck to Roxy and Renata. Also good luck to Rachel, Oscar, and Nor. Good luck to Nada, Miss Johnson's AP Bio class, Miss Churchill's AP Bio class, Nick Nor. Also Miss Brick's AP Bio class, Zeneb and Stephanie Z. Good luck. Yash. Also to Wendy, good luck. And Tiffany, good luck. Also to Thomas, Terrence, Telvin, Sierra, Sumia. So that was a lot of folks from California. I hope your state does well on the AP exam. From Colorado, good luck to Alex, Kelsey, Kelly, Sanya, Sanyana, Brad, Sarah, and also Ephraim. Good luck to Fran as well from Colorado. In Connecticut, please, good luck to Miss Balazzi's AP Bio class, to Jasmine, Carolyn, Ank, Lauren, Eliza, Tyler, Kate, Isabella, Darwin's Finches, sounds really cool. Dan, also good luck to Dr. D's AP Bio class, Jade, and Pasquale. Next, in Delaware, good luck to Mike, Lily, Srini, Ian, Frank, and Adam. From Florida, good luck to Shreni, Rachel, Caitlin, Jacqueline, also Marion, Elizabeth, Gabby, and Sarah. Good luck to Megan, Maria, Julie, Arcredi, Lauren, Carrie and Carly, Ellie, good luck, Jordan, Joshua, Sigil, Emily, Vernica, Dr. McMaster's AP Bio class, to Joey, good luck, Parker and Miss Malone's AP Bio class, also to Desperate Help, good luck, Hillary, Abigail, Juan, Ann, and Austin. Wow, that's a lot of people from Florida. Good luck as a state. In Georgia, good luck to Jen, Harley, Josiah, Zara, Dane, and Mike from the great state of Georgia. From Illinois, good luck to Cameron, Lydia, and Kavita. From Indiana, good luck to Bill and Southmount High AP Bio class. Good luck to Dan and Rachel and Cora. In Iowa, good luck to Nancy, Sarah, Tim, and Tim. In Kentucky, good luck to Sabrina, Jackie, Kevin, Sarah, Miss Smith's AP Bio class, and to the Henderson County AP Bio class. Good luck. From Maine, good luck to Kate and Hannah. In Maryland, good luck to Amanda, Jules, Jack, and Mr. Mazzella, Sierra, Tim, Ashton, Matt, Chandler, Brody, Brandon, and Molly. Good luck to Kiki, Mr. Scott's AP Bio class, Sahana, Chigo, and Jamima. Also, in Massachusetts, good luck to Mrs. Doyle's AP Bio class, Mrs. Tom's AP Bio class, Mrs. Lygick's AP Bio class, to Stephen, Mr. Walsh AP Bio class, Megan. Also, good luck to Maddie and Lavender and Lauren and Kennedy and Miss Rayner's AP Bio class. Good luck, Julia, Heather, and Ellen. And also to Brester, Jared, Philippine, Monica, Tian, Haram, and Russell. Also, good luck to Ashley, Belmont AP Bio class, and finally, Abigail. In Michigan, good luck to Israeth and also Duris, Alex, Kayla, Mike, Brandon High School AP Bio class, to Haley, good luck, Harris, Coruna AP Bio class, Muhammad, Piper and Annie, Kevin, good luck, Neemin, Raymond, Ricardo, and Sarah from the great state of Michigan, good luck. Minnesota, good luck to Thomas, Abel, probably, Martha, and Lily. In Missouri, good luck to Mrs. Ketchum's AP Bio class and to Lakshmi. Next, in Montana, good luck to Mrs. P's AP Bio class, to Nina, and Mr. Anderson's AP Bio class. Good luck, guys. I hope you do well on Monday. Nevada, good luck to Marisol, Jocelyn, and Cassette. In New Hampshire, good luck to Mrs. Grimsley's AP Bio class. In New Jersey, good luck to Burrell. Also to Erica, Amel, Barbie, Ari, Nicole, and Rajani. To Daniel, good luck. Jennifer, Rachel, Kate. In New York, good luck to Amanda and Damien. To Dylan, to Chloe, to Abby, to Alyssa, to Amira, to Brandon, to Ang, to Christopher, or maybe that's Angie, to Justin, to Latal, to Miss Rogers' AP Bio class. Good luck to Rohit, to Rosie, to Yael, to Louie, to Kyle, to Kulsuma, to Liam, to Lewin, to Highland, to Emily, to Aaron, to Fazil, to Danielle, good luck, to Damien Am, to David and Amy, to Julie and John and Loyola AP Bio class, to Iki, to Ike, to Jack, to Sky, to Amanda, to Tiffany, to Victoria, good luck, to SpongeBob, to Kareem, Brennan, and Jared, to Emily, Alex, Alexa, Chloe, Peter, Megan, Danny, and PJ. Also good luck to Mrs. D's, Mr. D's AP Bio class. Good luck to Miss Slomnicki's AP Bio class, and to Naomi, and also good luck to Sid and Sarah, Ricky, and Monica. A lot of people in New York. Good luck, guys. In North Carolina, good luck to Matthew and Desmarais. Also good luck to Autumn, to Todd, Cora, Jasmine, Erica, Kendra, Ashley, Taylor, Victoria, Mackenzie, and Meredith. Good luck to Juan and Heidi, to Miss Hinson's AP Bio class, to Jake and Paniota, to Joshua, to Haley, to Reagan, and Jess. In Ohio, good luck to Mr. Bunce's AP Bio class, to Alyssa, to Miss Abrams' AP Bio class, to Tiffin Columbian Biology class, or club, wow. To Nate, to Laura and Ashley, Courtney, and Rachel, good luck to Drew. In Oklahoma, good luck to Otson and Lottie. In Oregon, good luck to Reese. In Pennsylvania, good luck to Queso, Carolyn, Ryan, and Michael. In Rhode Island, good luck to Emma and Victoria. In South Carolina, good luck to Shashank and the SHS AP Bio class. In South Dakota, to Miss Rydell's AP Bio class and to Alex. In Texas, good luck to John and Emma and Ibrahim, I think that's right, to Christy, to Daisy, to Hill County Christian AP Bio class. Good luck to Amy and Alexandra and Shweta and Becca and Sunny and Caroline and Miss Reedy's, Mr. Reed's AP Bio class, and Mrs. Rice AP Bio class, and Rowan and Ruby and Mrs. Abney's AP Bio class from the great state of Texas. Good luck, guys. In Utah, good luck to Gianni and Mark and Mr. LeDuc's AP Bio class. Good luck. In Virginia, to Nikhil and Sydney and Ashley and Annie. In Washington, to Jude and Grace and Michaela and Sarah and Mrs. Lefstad's AP Bio class. To Kyle and Alan and Julia. In West Virginia, good luck to Jason. In Wisconsin, good luck to Angela and Katie and Sarah and Nicole and Sri and Alex. Now we're going to the countries. In Austria, good luck to Gade, and in Canada, to Taya and Sam and Selena and Nudrat and Masut and Lyndon and Kathy and Jessica. From Hong Kong, good luck to Rachel. In India, good luck to Rapali. In Italy, good luck to Santiago. In Jamaica, good luck to Zachary. And in Mexico, good luck to Bria and Natalie. And in the Netherlands, good luck to Molly. And in Pakistan, good luck to Farouk. And in Puerto Rico, good luck to Alexis. In Saipan, good luck to Ashley. And in Saudi Arabia, good luck to Shua. In South Korea, good luck to Allison. And in Switzerland, good luck to Marcus. And in Taiwan, good luck to Monica. Thailand, good luck to Sally. In Trinidad, good luck to Miss O'Donovan's AP Bio class. And in the United Arab Emirates, good luck to Ryan and Mohammed. And in United Kingdom, good luck to Efe and Hytham in the U.S. Good luck to Kieran and Hope and Joshua and Katie and Coral and Kelly and Ariana and Bella and Albert and Jenna and Mayura and Michelle and Tyler and Peter and Nikhil and Will and Yanni and Tina and Samir and Ralph and Jimmy and Harry in Venezuela. Good luck to Camilla. Wow, I'm done. So good luck to all of you. I hope you do really well on the AP exam. I hope all the videos have been helpful. My voice is gone. I'm tired. And so good luck to everyone. And I hope all of this was helpful.